Hey guys, my name is Matthew Olszewski.
I'm Olszewski.
And we're going to talk to you about our project, which is biochemical analysis of the MAPK pathway during fertilization of blastogenesis in the sea urchin Lytechinus species.
So sea urchins have a unique cell signaling pathway. Unlike other species, when MAPK is present in sea urchin signaling pathways, the cell cycle goes through a unique arrest called a G egg, and when MAPK is not present, the cell cycle goes through its normal phase, and we believe this is contributed to by the MOS protein. So our methods, we were collecting local urchins from places such as Fort DeSoto. We would then bring those urchins back to our university where we have a unique aquaculture tank room set up. We then used those urchins to collect sperm and eggs, which we were then able to use for protein analysis.
And then I took those protein lysates and I loaded them into SDS page gels and ran those, separating the proteins by size. I then transferred those to a Western blotting membrane and performed the Western blot analysis on those. So over to the results panel, you can see in the top figure, figure 1, that is the SDS page gel stained for total proteins. Going left to right, you have your ladder, your unfertilized eggs, and then 2.5, 5, 10, and 20 minutes post-fertilization. And you can see that all of those black bands look pretty similar. Those black bands are just showing that's the protein itself. And then the figure 2 and figure 3, those are the Western blots. The figure 2, that Western blot was done previously in the lab, and that is showing the presence of proteins, again, at the unfertilized eggs, and then 2.5, 5, 10, 20, and so on. The top row is showing MOS, and then MAPK beneath that, and then MAC in the very bottom row is basically a control. And then the Western blots that we ran, we were looking for phosphorylated MAPK and then the total MAPK. And as you can see, all of those bands are pretty dark. So that indicates to us that MAPK, both phosphorylated and total MAPK, remain pretty high, at least up through the 20-minute post-fertilization mark.
Some future directions we have for this project are we hope to increase the time replicate so we can continue running separate Western blots and get more results. And we also want to identify new time points that we could run that kind of focus on the MOS and MAPK pathways present in the eggs.
And then we would also like to expand on Western blotting using different antibodies to include other protein targets. We would like to image the cells live, being able to actually see the chromosomes separating and dividing. We want to do some inhibition experiments of MAPK to see what would happen if MAPK was not there. And then lastly, we would also like to see what would happen if we were to introduce MOS into eggs, doing a single blastomere sufficiency test with those. So thank you guys for coming to our presentation. If you have any questions, let us know.